Hello, good evening, good afternoon, good morning, wherever you are. This is David Burger, clinical pharmacist and clinical pharmacologist from Radboudumc and also from Global DDI Solutions. And I'm happy to discuss with you the drug-drug interactions as a manageable risk when applying oral antivirals for COVID-19. So here are my disclosures. So what I'm going to discuss with you today is the drug-drug interaction risk of currently available antivirals for SARS-CoV-2. And that will mainly focus on ritonavir as part of these antivirals as a perpetrator of a DDI. But however, ritonavir can also be a victim of a DDI. All that knowledge that we have on drug-drug interactions with ritonavir will lead to the management of the drug-drug interactions in COVID-19 patients, which I will explain to you. And I will finish with some conclusions. So when we talk about the drug-drug interaction risk of SARS-CoV-2 antivirals, we are not talking about the monoclonal antibodies and also not on molnupiravir, another oral antiviral, because these agents do not cause significant drug-drug interactions. We are mainly talking about the agent nirmatrelvir boosted by ritonavir, also known as Paxlovid, because that combination has a very high potential for clinically relevant drug-drug interactions. But maybe some of you will be scared about drug-drug interaction, that it's always bad news when there is a drug-drug interaction. But my statement is that drug-drug interactions are not dangerous. The only dangerous drug-drug interaction is the one that is missed. All the others, I think we can manage them. And I'm sure that after my presentation, you also will feel more confident in management drug-drug interactions. And often that needs good communication between the physician, the prescriber, the pharmacist as a dispensing, and also the patient. So when we talk about the drug-drug interaction risk of nirmatrelvir boosted by ritonavir, we are mainly talking about ritonavir because that's added to nirmatrelvir to boost the plasma concentrations of the antiviral agent. And in that situation, then the nirmatrelvir, which is the active agent, becomes the therapeutic agent. When you give the nirmatrelvir only without ritonavir, it has a very short half-life, elimination half-life of only 2 hours. And if you would give that to a patient, maybe you should give it 5 times or 6 times a day to achieve any efficacy. However, when you combine it with ritonavir, you are boosting the concentrations and the half-life is extended to 7 hours, and you can give the drug 2 times a day. And that effect of ritonavir boosting nirmatrelvir is based on CYP3A inhibition, where the nirmatrelvir is the substrate, the CYP3A substrate, and ritonavir is the inhibitor. And this concept is not new. In fact, we are celebrating, I would say, this year, the 25th anniversary of the concept that was originally developed by Dale Kempf and colleagues from Abbott when they were looking at ritonavir, which was originally developed as a therapeutic HIV protease inhibitor, but now using it in a lower dose to increase the plasma concentrations of other HIV protease inhibitors. And that concept was called pharmacokinetic enhancement, as you can see in this paper. These two graphs are the first 2 patients where this concept of boosting by ritonavir was applied, a male patient and a female patient that were treated with saquinavir, the HIV protease inhibitor, which, as you can see, only achieves its very low concentrations, which are eliminated quite rapidly. However, if you combine it with ritonavir, you get largely increased concentrations of saquinavir. And please look, this is a logarithmic scale. So you have almost a 50-fold increase in exposure to saquinavir when it's combined with ritonavir as a booster. If you want to know how this exactly is happening, you can have a look at this slide that I received from Professor David Beck, which explains all the various ways how you can have a drug-drug interaction with an orally administered agent. So the drug is being administered, is transported through the gut, and then it is absorbed through the enterocyte into the portal blood. Then it's being uptaken in the hepatocyte, and then either it's transported back to the portal blood or it's excreted in the bile duct. And you see there are various ways where this process can be influenced and how a drug-drug interaction may be caused. If you then look at what ritonavir is doing, actually, it has an effect on all of these different mechanisms. So it's influencing the uptake into the enterocyte by inhibition of PGP. So you get higher levels that's being absorbed. It's also influencing the uptake into the hepatocyte, and it's also influencing the CYP enzymes that are present in the hepatocytes. The net effect of all these mechanisms is usually a higher concentration of the other agents, which can lead to either more efficacy or to more toxicity. So you not only have to know the mechanism of that drug-drug interaction caused by ritonavir, you also have to look at the exposure-response relationship, because sometimes small increases in drug levels do not give a significant difference in effect or toxicity, and sometimes they do. So you not only have to know the mechanism, but also the impact in terms of the exposure-response relationship. So ritonavir as a PK enhancer is not new, as I mentioned. We have more than 25 years of experience, and as we speak, millions of people worldwide are now using a boosted agent, boosted by ritonavir. So this is definitely something not new, and we have a lot of experience with it. And not only in HIV, maybe some of you remember that when the first wave of direct-acting antivirals came for hepatitis C treatment, we also had the combination product Vigirax, where also ritonavir was used as a booster. And in fact, there has also been a drug specifically developed as a booster named cobicistat by Gilead, which has the sole indication of pharmacokinetic enhancement, and it's included in many single tablet regimens such as Genvoya and Simtusa and also others. You can see the differences between cobicistat and ritonavir explained in this paper. So you can imagine that if ritonavir is added to increase the levels of the therapeutic agents, it may also increase levels of co-medication. And then we are talking about a drug-drug interaction that might be unwanted. And that's in particular relevant when a drug is a CYP3A substrate and also has a narrow therapeutic range, because then the increased levels may lead to more toxicity. And that has led to a number of contraindicated CYP3A substrates that are mentioned here in the product label of Paxlovid. It must be noted that this is only a list, not a complete list of all possible CYP3A substrate, but it already gives you a good overview what kind of agents cannot be used at the same time with Paxlovid. Another aspect that is important to realize is that this effect of CYP3 inhibition is already present directly after the first 2 or 3 doses of ritonavir. When this Paxlovid treatment of 5 days was developed, some people thought, well, only 5 days is not really clinically relevant to cause a drug-drug interaction. But that's not true. You can really see, and some case reports have already shown that in the past in HIV patients, that after the first doses of ritonavir, you can have increased levels of your CYP3A co-medication, leading to clinical toxicity. So it is relevant also with a short course. Also important to understand is that partly the mechanism of CYP3A inhibition by ritonavir is inactivation, which means that as a result, the lower CYP3 activity may persist for a few days after ritonavir is stopped, because it takes some time before new CYP3A is synthesized. So that's in another 2 or 3 days that there may be impaired CYP3 activity. That actually might even be longer in some individuals. As a practical advice, we normally say that the risk for drug-drug interaction is present with Paxlovid during the 5 days of its use, plus an additional 3 days, which makes it 8 days. So if you would start Paxlovid, let's say on the 15th of June, then you could say on Wednesday, 1 week later, the interaction will be less relevant or will be over. So just as a practical advice to your patients. However, this is not a full story. We're not talking only about CYP3A inhibition, because ritonavir does more. It is also a weak inhibitor of CYP2D6, another isoenzyme of the CYP450 system. Normally, we do not pay a lot of attention to that drug interaction because it's only a minimal increase. However, it could be relevant in certain situations where you have a CYP2D6 substrate with a narrow therapeutic range, such as certain antiarrhythmic agents. And also antipsychotics can be CYP2D6 substrates, so those should be cautioned when combined with Paxlovid. Furthermore, ritonavir is a mixed inhibitor, but also inducer of the membrane transporter PGP. And studies have shown that if you use it chronically, you initially get PGP inhibition, then followed by PGP induction. And a study with Drugdrug showed that after 14 days, the net effect on PGP is neutral, so not clinically relevant. However, a recent study that was included in the updated label of Paxlovid showed that 5 days of Paxlovid with dabigatran showed about a twofold increase in the levels of dabigatran, which is clinically relevant. And it could also be relevant for other PGP substrates that have a therapeutic range, such as digoxin or certain anti-cancer agents. And then finally, ritonavir can also induce some other enzymes like UGG and CYP2C9. However, we feel that with the short duration of 5 days of Paxlovid, that is probably of low clinical relevance. That may change if maybe in the future, longer periods of Paxlovid use are going to be studied and recommended. But for now, we think 5 days is not relevant to cause significant induction. So far, I've been talking about Paxlovid as a perpetrator that causes a DDI. People not always realize that also the Paxlovid combination can be a victim of a DDI, so that other agents are influencing the concentrations of the oral antiviral agents. And that is not very surprising because both nirmatrelvir and ritonavir are CYP3A substrates. So if you combine that then with a strong CYP3A inducer, such as rifampin or certain antiepileptics, then you get a lower concentrations of the oral antiviral agents, and the booster is no longer present anymore. So that's important also to realize. And then finally, in HIV treatment, we do have some experience that you can overcome this induction by doubling the dose of the ritonavir boosted agent, or sometimes you can add extra doses of ritonavir to compensate for the induction, but we don't have any data for Paxlovid to do that. So now we have all the knowledge of the drug-drug interactions that can either be caused by Paxlovid or that when Paxlovid can be the victim of a DDI. How are we going to translate that into a DDI management? Well, then we can make use of the very helpful tool developed by the University of Liverpool. I hope many of you already have experience using that tool. Otherwise, I would recommend to do it. Where you can add the nirmatrelvir ritonavir combination with any of the 630 co-medications that already listed, and you get an advice how to combine this medication. What is important if you talk about this proactive management of a drug-drug interaction, which means that before you're actually prescribing or dispensing the co-medication, you already can have a recommendation how to manage a potential DDI, is that in the case of the short course of 5 days of Paxlovid, we now have an additional option in our DDI management, which we normally not have in when we are using ritonavir chronically as a boosted, because now we can think of temporary discontinuation of the co-medication, which of course is not possible if you have chronic treatment. For instance, if you look at two of these examples, when you talk about statins, we normally say that it's not very harmful for a patient if he's not taking the statin for a few days. So in this case, to avoid a drug-drug interaction, one of the most easiest intervention is to stop the statin for this period of 8 days. And also with tacrolimus, which we know there can be very high increases in levels of tacrolimus when you combine it with ritonavir. If you stop it, then still tacrolimus may be present at therapeutic levels for at least part of this period of 8 days. And that's also now recommendation by our colleagues from the transplant world. And in this case, when you in fact have to answer two important questions. So one question is there a significant interaction? Yes. And also the other question is, the communication cannot be stopped. So only if you have the answer yes to both of these questions, then you really have a true contraindication for Paxlovid in such a patient. And there are some examples where indeed there is a strong interaction and indeed you cannot stop it for 8 days. And then there's a true contraindication. So this is all listed in more detail in what we call this DDI algorithm, that was presented in this paper earlier this year by Katia Marzolini, where in fact you can look at all different co-medications and lead them through this algorithm, where you can have various situations. For instance, if there is no DDI expected, you can safely give the Paxlovid combination. And also when there is only a weak DDI, because of the short duration, we do not think that's a big problem. And only in a very exceptional situation where you have a strong interaction, you cannot continue the co-medication, then maybe you should consider another treatment for Paxlovid and better not give it. But in all the other situations, if you know how to do it, you should safely give Paxlovid to these patients. So when we are focusing about these drug-drug interactions, there is a risk that we only think about drug-drug interactions so that we develop some kind of a tunnel vision. But we also have to look, of course, at other factors. It may be different if the patient is underweight or heavily obese. And also, for instance, if there is a renal dysfunction in a patient, because the management of certain drug-drug interactions may be different in a patient with a normal renal functions versus a patient where there could be renal impairment. And this is just an example of a direct oral anticoagulant agents where you maybe have a different management of your DDI in case there is also impaired renal function. So coming to a conclusion, I hope that you agree with me that with good knowledge, when we understand the mechanisms, that the DDI management is not difficult. We really have the knowledge how to do it. Of course, it does require some proactive communication between various healthcare professionals and, of course, the patient, him or herself, but we can do it. And I'm coming back to this important slide. And now I hope you are feeling more confident that DDIs are not dangerous. The only dangerous is if you miss it. But based also on what I told you today, I truly believe that all others can be managed. And with that, I thank you for your attention, and I'm sending you the greetings from lovely Nijmegen. Have a nice evening.